compounds. These bacteria play a critical role in what's called bioremediation, breaking down toxic chemicals that usually humans put together and dumped in the environment someplace, and then we can use these bacteria to get rid of them. So Pseudomonas are very important for that function. Anyway, this is the essence of what we were doing for our Pseudomonas enrichment. Right now, our Pseudomonas enrichment medium is broth. So it's over here on the shaker. And what you would notice if you look at these Pseudomonas media tubes, as they're on the shaker, now there's a layer of bubbles. That bubbly material indicates we've got some biofilm formation. So organisms are growing in this. And what I'm going to do with these is take these bubbly materials and put them on plates. Ordinarily, you guys will do this. You don't get to do it because we don't have the medium. So medium is being made right now. It will be available tomorrow, and I will transfer these for you guys. So I hope your tubes are nicely labeled so I can pull the label in the plates and transfer this out. You can say, well, why don't you let us do it next Tuesday? Because they shouldn't stay on the shaker too long. They've been on there probably a little longer than they should have been. I don't want to leave them on till Thursday. Okay, our next enrichment is the Azotobacter enrichment. This medium has no nitrogen in it. Nitrogen is essential for bacteria to make proteins, nucleic acids, peptidoglycan. They have to have nitrogen to do that. So if the medium doesn't have any nitrogen, it's going to inhibit the growth of any bacteria that can't fix nitrogen. What we mean by fixing nitrogen, some bacteria can take N2, molecular nitrogen from the atmosphere. They initially make nitrate and ammonia with it, but eventually they can form organic compounds. Azotobacter can do this. They can fix nitrogen. Turns out they're not the only organisms that can, but they are one of the ones that can. So this medium, no nitrogen in it. Bacteria that can't fix nitrogen are not going to grow. But Azotobacter can, because they fix nitrogen. The last enrichment that we did involved treating our soil in a particular way. So you put some soil in a tube with water, and then you pasteurize it. Pasteurization involves applying heat, specific temperature for a specific period of time. We had a boiling water bath going up here. When water boils, it's about 100 degrees centigrade. So boiling, we put it in there for 1 minute. So they boiled. That boiling is going to kill vegetative cells. It'll kill the Pseudomonas, it'll kill the Azotobacter, it'll kill whatever else is in there. A vegetative cell is the living, growing cell type. What it doesn't kill are endospores or exospores. And the reason those don't die is because they are thermoduric. This term means they can endure heat. It doesn't kill them. So they're highly resistant to heat. Tonight I talked about endospores, these dormant structures. They have 2 layers of membrane, 2 layers of wall. They contain almost no water, so they're metabolically inactive. They're extremely resistant. So we can boil them probably longer than 1 minute, and they wouldn't die. And then what you did was take your liquid and stick the nutrient agar. The spores will undergo germination on the nutrient agar, and they will grow new cells. So that is how we did our enrichment procedure for bacillus. Bacillus get to survive because they are spore formers. I should write that on there. So the bacillus are endospore forming bacteria. They're one of the genera. So this is our soil enrichment. Again, this is an assignment that is going to be due out there a couple of weeks. But I wanted you to get the gist of what is required for this, because we just keep having new assignments. They just keep piling on. So get this stuff recorded. The other thing that we're doing tonight is hopefully finishing up this exercise where we were looking at the isolation of a pure bacterial culture. So last lab, you looked at Staphylococcus and Klebsiella on the mixed egg plate. Serratia, Marcescens, and Bacillus cereus on the mixed feed plate. And your job was to select one of those 4 organism types and streak a new plate. Your new plate had to have the proper label. It had to be Staphylococcus aureus or Klebsiella pneumoniae or Serratia marcescens or Bacillus cereus. You couldn't call it mixed A, mixed B because it wasn't going to be. It was going to be a pure culture. A pure culture contains only one organism type. Your colonies need to be well isolated. So where I say it has to be a pure culture, it has to be properly streaked. That means there have to be isolated colonies. I had some students from, I think it was Sasha's class, and they said, well, I've got one isolated colony, does that count? I'd like you to have more than one. You should have multiple. The reason for getting isolated colonies is that allows you to see if the culture is indeed pure. If you don't have isolated colonies, you can't tell. You won't know. So you need several isolated colonies. And then you might be thinking to yourself, do I really need to learn how to do this technique? In this lab, you do. Because physiological unknown 1 is going to come to you as a block with 2 different organism types. And you have to be able to streak and get isolated colonies and separate those guys. Otherwise, you can't identify them. Okay, so this is a technique you need to learn. You need to learn how to do it, how to streak to get isolated colonies. So if you have a culture that is indeed pure, it's properly streaked, you have isolated colonies, and it is properly labeled, again, your name, the date, the medium type, the culture ID. You can't say mix A, mix B. If it fits those criteria, turn it in. It's gonna go right here on the front bench. I clean this already. I just put the streak. If it's not, you look at it, you go, no isolation. Or you look at it and go, that's not pure at all. I've still got Bacillus and Serratia. Just do it again. So the media are out here. TSA, dripping soy agar, this is for Klebsiella or Staph. Nutrient agar, this is for Serratia or Bacillus. Your plates, mostly they're in your drawers. I can look in the box and see if I put up any of them in the box.
Look, I grew stuff. Look, I grew stuff. Oh, that's yours. I know. So which one is my which one is my
Did you label it up? Because I label mine unknown.
I did
And then
not.
number 26. You didn't write it?
This is your
Oh, there it is.
No, we're not doing the morphological unknown.
We're not doing morphological unknown.
No, we're doing the.
Serratia?
You need your lab coats on.
This one?
We are
Yeah.
working to mix A, mix B. Remember, our mixed A cultures are potentially pathogenic. If you left your plates in the incubator on Thursday night, they are over here on the side bench. So what's over here is Staphylococcus and Klebsiella.
Did you really? On this? I don't know, my shit sucks. Yeah, ours is the same. We do it the same way.
Mine's all crazy.
I think that's how it's supposed to be, though.
I feel like everybody does it like this.
Yeah.
DJ, where'd you open it at?
Yeah, I think everyone in here is just anal.
I'm just